Hello students, welcome to our video tutorial class. We all know that before the vacation we have already completed up to the second chapter of the first unit, that is the sexual reproduction in flowering plant. So today in this video I am starting the third chapter, that is human reproduction. Keep it in your mind, this chapter is very much important for our boards exam as well as who are appearing for the NEET. For them also this chapter is so much important because so many questions are coming from this specific chapter. So let's start the new chapter, human reproduction. So first of all, when we will be starting with the human reproduction, first what we need to know? We need to know that the humans are sexually reproducing organisms and they are commonly known as the viviparous organisms, because this specific idea we have that humans are sexually reproducing organism because they can't follow any other kind of reproduction mechanism, as well as they are producing their own baby. They are not producing any egg, and from that egg they are producing the next generation individual. It's not the mechanism for them. Obviously they are producing directly the baby within the mother's womb. That is the main thing in case of human. So the reproductive events which are happening in human, that is including some of the steps. So let's see the steps. First step is gametogenesis. From the name we can understand gametogenesis means the formation of gametes. Gametes means obviously male gamete and female gamete, that is sperm and ovum respectively. So always the first step will be the formation of the male and female gamete. Okay. Second step, insemination. That means after formation of male gamete and female gamete within the male and female body respectively, the introduction of the male gamete, that is the sperm, within the female reproductive tract, and that is inseminated, or that will be introduced through the semen, that is a whitish colored fluid, that is the medium for sperm entry, or that is the medium for the movement of the sperm, through which the male gamete, that is the sperm, will be getting transferred from the male individual to female individual's body. Third step is fertilization. That means when the male sperm is already introduced within the female body, then there is a specific location in the female reproductive tract where the sperm and ovum, they are fertilizing with each other. That means they are fusing with each other. Okay. Next, after fertilization, the next step is coming, that is formation of zygote, and from the zygote formation the next thing is coming, that is blastulation. So keep it in your mind, when the fusion has occurred, the fertilization has occurred, obviously that is forming the zygote, and after formation of the zygote, that zygote will be entering within the cell divisional steps, and it is further developing into the next stage of embryonic development, that is called as blastula. After formation of blastula, that blastula will be get attached in the inner wall of uterus, which is commonly known as endometrium. So when it will be getting attached to the inner wall of uterus, then it is commonly known as implantation. After implantation, obviously the embryonic development, it will be starting in a higher speed, or rather you can say the actual developmental process of the embryo, it will start when it is getting adhered to the inner wall of uterus, that is endometrium, and this embryonic development will take place for 270 days, or that is 270 days, 9 months, almost a 9 month 10 days. So this entire time period of embryonic development is commonly known as gestation. After gestation, you can say that the maturation of the fetus, that is taking place, and that is actually responsible for the delivery of the baby, and when the baby delivery or the fetus delivery it will take place, that is commonly known as parturition. So after gestation, the delivery of the fetus or the delivery of the baby which takes place, that is commonly known as parturition. So I think in these two slides, whatever the steps of reproduction in human I have told you, that is gametogenesis, insemination, fertilization, blastulation, implantation, gestation and parturition, I think these all steps are very much clear to each and every student. So we are going for the next slide. We are proceeding. In the next slide, we are starting with specifically the male reproductive system. So we will be discussing at first about the male reproductive system, and in the next video we will be discussing about the female reproductive system. So first of all, in the male reproductive system, we need to know about the portions or the segments of the male reproductive system. Whatever the portions, in the male reproductive system has been segregated into 4 parts. One is male reproductive unit, that is commonly known as testes or testis. Testis is the singular and this is actually written testes. Testes is the plural. Okay. Next, reproductive tract is also present. Third, there is a presence of male reproductive glands which are supporting in the reproduction procedure, that is responsible for producing the medium of sperm movement, that is the semen. And the last one, that is the male external genitalia, which is commonly known as penis, and that is responsible for introducing the sperm within the female reproductive tract. Now see, in case of reproductive tract, that is segregated into multiple portions, because reproductive tract in case of male, that is a lengthy portion. So whatever the parts can be found in case of reproductive tract, first part, that is the retetestis. Though this retetestis portion is present within the structure of the testis, but from this retetestis portion is actually the reproductive tract structure is starting. So commonly in our books it will be actually written the reproductive tract is starting from epididymis. It's not starting from the retetestis. But if you want to include this retetestis within the reproductive tract, it will not be wrong. So the portions under the reproductive tract will be first one retetestis, second one epididymis, third one that is a vas deferens or ductus deferens, fourth that is the ejaculatory duct, and fifth that is the urethra. So one by one, each and every portion of reproductive tract, what are the male reproductive glands, male external genitalia, what is that and what is their structure, what is the structure of testis, these all points we will be discussing in the next slides. So we are just proceeding for the next slide and we will be starting there about the discussion of the male reproductive unit, that is the testis. So first of all, you can see the overall diagram of the testis. This is the overall structure of the testis. So let's see whatever the structural parts are commonly found in case of testis. First one, that is obviously the septa. Septa is obviously the membranous structure which are actually separating the entire testis into multiple lobes or lobules. So for that reason, lobules are also written. Those are commonly known as testicular lobules. Within testicular lobules, seminiferous tubules are present, which are actually the production house of the sperm. Then straight tubules are also there. Then efferent ductules are also there, and further retetestis is there. So here you can see, already I have told that the retetestis is actually kept or commonly kept within the testis. But if you want, you can keep within the reproductive tract, or if you want also, you can keep it within the testis portion. No problem. Okay. Now just we are proceeding for the next slide, that is the structure of the testis. But before that, we will be discussing about one small portion, that is the scrotum or scrotal sac. What is that? Here it is actually a scrotum or the scrotal sac is actually a sac-like structure which is located behind and below the penis. Keep it in your mind, this is a small muscular sac-like structure which are generally containing as well as protecting the testicles or testes. That means the testes, if anybody will ask you where actually the testes are present in male body, obviously your answer will be it is present within the scrotum or scrotal sac, and that sac-like structure are present on the both sides of the penis and towards the back side of the penis or male external genitalia, and both of the sacs are present in hanging condition. Inside the scrotal sac there is a cavity, and the cavity is named inguinal cavity. Keep it in your mind, the cavity within the scrotal sac or the scrotum, that is commonly known as inguinal cavity. Keep it in your mind that within the inguinal cavity of the scrotal sac or scrotum, the temperature is 2 to 3 degrees Celsius lower than the normal body temperature. Why? So that the spermatogenesis can happen within the testis, because that temperature is favorable for spermatogenesis. Keep it in your mind, the formation of the sperm which is taking place within the seminiferous tubule of testis, this specific spermatogenesis process is not happening in our body temperature, because our body temperature is not suitable for spermatogenesis process. Rather, the temperature which is present within the scrotal sac, that is 2 to 3 degrees Celsius less than our normal body temperature, that is quite favorable than our body temperature for the spermatogenesis. So for that reason, the spermatogenesis is always taking place inside the scrotal sac due to presence of 2 to 3 degrees Celsius less temperature from our normal body temperature. Now just we are moving for the next portion, and the next portion is the structure of the testis. This is the overall structure of the testis. You can see here, you can see this is the entire testis structure, which is subdivided into multiple lobes or lobule. Here it is written, and the lobules are subdivided, or they are separated from each other by the help of a membrane that is called as septum. Okay. Now the point is, within the septum you can see the coiled tube, and those coiled tubes are commonly known as seminiferous tubules. Next, these seminiferous tubules are joining with the capillary-like structure. This is the capillary-like structure, whatever I am showing in the diagram, that is commonly known as rete testis. And this specific seminiferous tubule are joining with the rete testis by a small straight duct-like structure that is known as straight tubule. Keep it in your mind, from the rete testis again some straight duct-like structures are coming out. Those are commonly known as efferent ductule, or it can be also called as vasa efferentia, and this vasa efferentia is connected with the entire structure. This portion I am talking about, this is known as epididymis. This epididymis portion is connected with the next portion, that means this portion I am talking about, this is known as vas deferens, and this vas deferens is entering within a cord-like structure or a tube-like structure, which is also containing blood vessels and nerves. So blood vessel, nerve and the vas deferens portion, these three are collectively present within a duct, and that is commonly known as spermatic cord. And overall the testis is covered by a membrane, that is tunica albuginea, and with that tunica vaginalis, tunica vasculosa, this kind of layers are present. But according to our board syllabus, we will be discussing only that the testis is covered or surrounded by a membrane, that is tunica albuginea. So let's see the structure of the testis. Each testis is oval in shape. From the figure, this figure we can understand obviously that is an oval shaped structure. Next, each testis is covered by the membranous covering, and that is called as tunica albuginea. You can see here tunica albuginea is the covering of the testis, and outside of the tunica albuginea, tunica vaginalis is also present, but that is not mentioned here because tunica vaginalis is not so much important for our board syllabus. Next, entire testicular tissue is subdivided into multiple lobe-like or lobule-like structures. You can see here, and I have already shown you in this slide, the entire testis structure is subdivided into multiple compartments or lobules. So number of lobules are almost 250 to 300 within each and every testis, and those lobes or lobules are commonly known as testicular lobules. Keep it in your mind, these lobes or lobules are commonly known as testicular lobules. Next, each testicular lobule is separated from each other by a membrane-like structure that is known as septa. That also I have told you, this is the septa or septum. If it is single, on that time it is septum, and when multiple septum are present in between the testicular lobules, obviously the name will be coming septa. So the septa will be present for separating or differentiating the testicular chambers or lobules. Keep it in your mind, within each and every testicular lobe, coiled tubes are present, which are commonly known as seminiferous tubule. And keep it in your mind, these tubules are 3 to 4 in number. Okay. Next, seminiferous tubules come out from each testicular lobule by forming the straight tubule. Here, seminiferous tubule, you can see here already I have told this seminiferous tubule, which are coming out from the testicular lobule and forming some straight duct-like structure, and those are known as straight tubule. Clear? And those straight tubules are connected with a network of tubules or capillary-like structure, and that is commonly known as rete testis. So here also you can see rete testis is written within the testis structure, so you can write that, no problem. So I think the entire structure of the testis, which is related with this structure shown, it is very much clear to each and everyone. Now we are just proceeding for the next part, and the next part is male reproductive tract. That means testis structure we have already discussed properly. Now we have to know that whatever the duct or whatever the tubes are coming out from this testis and how they have reached up to the urinary bladder or whatever the location. So first of all, reproductive tract is composed of the following parts. Already I have told previously. First one, that is efferent ductule or vasa efferentia, then presence of epididymis, then vas deferens, then there is a presence of vas deferens also. Next, there is a presence of ejaculatory duct, and then there is a presence of urethra. So let's start from this specific slide where the first portion of the reproductive tract it is written, that is efferent ductule or vasa efferentia. What is that? Already I have told that the testis, within the testis up to the rete testis we have discussed. Now after rete testis, if you see this figure, from the rete testis again few straight duct-like structures are coming out, and these duct-like structures which are coming out from rete testis, this is the rete testis, that is a capillary network. So these duct-like structures are commonly known as efferent ductule or vasa efferentia. Okay. And keep it in your mind, by coiling these specific ducts, they are joining with epididymis portion. That means after vasa efferentia, obviously the epididymis portion is coming. So let's start with the epididymis portion. Epididymis portion, I have already told you, this is the entire portion which is the under the epididymis. This entire portion, whatever I am showing in the figure. So epididymis is the next duct portion which leads to vas deferens. Vas deferens is the next portion of the reproductive tract in the male reproductive system, and that ascends to the abdomen and loops over the urinary bladder. That means epididymis is the next duct portion after vasa efferentia, and this epididymis is connected with the vas deferens, and that is going to the abdomen and connected with the urinary bladder. Growth and development of the spermatozoa, that means when the sperms or spermatozoa are produced within this seminiferous tubule and they are coming out through the straight tubule and rete testis into the epididymis region, actually the spermatozoa are staying within the epididymis portion for a few times. Why? For their growth, for their further development. Okay. Next, epididymis is connected with the next coiled duct portion. This portion I am talking about, and this portion is commonly known as vas deferens. So let's start, or vasa differentia also it can be called. This is the next part of the male reproductive system after epididymis. The vas deferens is a long muscular tube that travels from the epididymis portion into the pelvic cavity. Pelvic cavity means the cavity or the portion which is present in our lower abdomen, pelvic region which is called, where the pelvic girdle is actually present, to just behind the bladder. Okay, the vas deferens transports mature sperm to the urethra. So that is the main function of the vas deferens, that within the epididymis the growth and development or the maturation of the spermatozoa takes place, and after the maturation gets over, then the spermatozoa will be transferred to the urethra by the help of this specific duct, that is vas deferens. Vas deferens, blood vessel and nerves are collectively present within a duct-like structure that is called as spermatic cord. Already I have shown you, this is the duct-like or cord-like structure which is commonly known as spermatic cord, and that is only going up to the urinary blood. So I think these three portions of the male reproductive tract, that is very much clear to each and everyone. Going for the next slide. This is the slide where it is written about the ejaculatory duct. What is that? The ejaculatory duct are the paired structure in the male. Each ejaculatory duct is formed by the union of the vas deferens with the duct of the seminal vesicle. They pass through the prostate and open into urethra. I am trying to show you what is the ejaculatory duct. Suppose this is the entire structure of urinary bladder. Okay. So from the urinary bladder, one duct-like structure is coming out. So the duct-like structure which is coming out from the urinary bladder and joining with the urethra, that specific duct-like structure is commonly known as ejaculatory duct. It is shown here also. This is the actual portion of the ejaculatory duct which is actually coming out from the urinary bladder. And one more thing, actually in the ejaculatory duct only it is actually formed due to union of the vas deferens, because vas deferens are getting united in this region only, as well as the seminal vesicle is also united with this ejaculatory duct for releasing its secretion. It means seminal vesicles secretion. And it is passing through the prostate gland, because these two, this one and this one, these two are the prostate gland. So it is passing through the prostate gland and opening into the next canal like structure, which is further passing through the male external genitalia, that is known as the penis. And that is the next part. Urethra. Urethra is a thin tube-like structure that begins at the lower opening of the bladder. That means the urethra is the next portion which is coming out from the ejaculatory duct, and it is actually starting when it has entered within the male external genitalia, and it is commonly known as external urethral orifice, and the canal like the aperture through which aperture the urethra is opening towards the outside, that is known as urethral orifice or urethral aperture. And keep it in your mind, this urethra is actually forming a canal like structure, and that is commonly known as urethral meatus. Okay, so this is overall the last two parts of the male reproductive tract, that is ejaculatory duct, that is actually coming out from the urinary bladder, the vas deferens as well as the seminal vesicles. Their ducts are all united in this ejaculatory duct so that their secretion can come out through it. And after that, the ejaculatory duct is connected with a canal like structure or the tube like structure which is passing through the male external genitalia, that is urethra. Urethra has two parts, one is canal like portion, that is urethral canal, and this canal is opening outside through a small aperture that is known as urethral orifice or urethral aperture, and the canal like structure is known as urethral meatus. Now just we are moving for the next structure, and the next structure is the structure of seminiferous tubule. I have already told you seminiferous tubule is a tube-like structure or the duct-like structure, and this duct-like structure is exclusively present within the testicular lobule, and this structure is exclusively responsible also for spermatogenesis. So let's start with the structure of seminiferous tubule. Already we know seminiferous tubule is a production house of the sperms. Each seminiferous tubule is lined by two types of cells. Always if you see the seminiferous tubule, if you transversely cut the tubular structure, you will see there are two cell layers. One is the spermatogonial cell layer, that is the outer layer, and next there is a presence of triangular shaped or conical shaped Sertoli cell layer. So spermatogonial cell layer, these cells are undergoing meiosis cell division to form the sperm, as well as Sertoli cell layer, that is provide the nutrition to the male germ cell or the sperm. Okay, so this is actually the structure of the seminiferous tubule. Next there is a point, that is the region outside the seminiferous tubule, that is called as the interstitial space. What is interstitial space? If you see the seminiferous tubules within the testicular lobule, except the cavity of the seminiferous tubule, there is a surrounding portion, and those portions are commonly known as interstitial spaces. And keep it in your mind, within the interstitial spaces some cells are also present. Those cells are commonly known as interstitial cells of Leydig, and these cells are exclusively responsible for producing the testicular hormone or the male sex hormone, that is the testosterone. Let's see this diagram, then it will be very much clear. This entire structure is actually the transverse section of the testicular lobule. Okay, it is the transverse section of the testicular lobule, so that you can see multiple seminiferous tubule. This is a portion of seminiferous tubule, this is the entire portion of seminiferous tubule, this is another seminiferous tubule, this is another seminiferous tubule, this is another seminiferous tubule. So many seminiferous tubules are present within the testicular lobule, so that's why these much cavities we can see. Keep it in your mind, in this pink region we can see so many cells, and these cells are actually interstitial cells of Leydig, which are actually present in between the seminiferous tubules. And keep it in your mind, these spaces are commonly known as interstitial spaces. Now if you enter within the seminiferous tubule, suppose I am considering this seminiferous tubule because it is entirely present, you can see peripherally located, that is the cells which are known as commonly spermatogonial cell layer, and after that there is a presence of Sertoli cells also. These are the triangular shaped Sertoli cells are also present. And keep it in your mind, these are the spermatozoa or sperm cells which are actually present almost centrally to the seminiferous tubule, and these spermatozoa only come out from the seminiferous tubule through straight tubule and coming into the retrotestis, and further it will be taking the pathway of the male reproductive tract and coming out. Keep it in your mind, after formation of the sperm or spermatozoa within the seminiferous tubule, the process of releasing of the sperm from the seminiferous tubule is commonly known as spermiation. Spermiation is the procedure by which the sperms are released out from the seminiferous tubule. So this is overall the structure of the seminiferous tubule, whatever we have learned in the previous slide. Okay, that means in this slide. Going for the next slide, because already we have discussed about the male reproductive unit as well as male reproductive tract, both of the parts. Next we are going for discussing about the male reproductive glands, and after that the last portion of the male reproductive system, that is the male external genitalia. So let's start with the male reproductive glands. Here you can see the male reproductive gland or accessory reproductive gland is mainly composed of three kind of glands. One is seminal vesicle, second one is prostate gland, and third one is bulbourethral gland. Here, keep it in your mind, the secretion of these all glands constitute the seminal plasma or the whitish colored milky fluid, that is semen, which is rich in fructose, calcium and certain enzymes, and it is acting as the medium for sperm or spermatozoa transport through the male reproductive tract, as well as after entering within the female reproductive tract also. This semen will only help to transport the sperm up to its destination, that means up to the ovum. Now we are just going for discussing about each and every male reproductive gland. First one, that is the seminal vesicle. In the seminal vesicle we can see that seminal vesicle is secreting the fluid that is constituting almost 60 to 70% of the entire volume of semen. Keep it in your mind, seminal vesicle is releasing the fluid which is alkaline in nature. Another most important point is seminal vesicle secreted chemical substance that is containing fructose and ascorbic acid, which are providing the nutrition to the sperm. And location of the seminal vesicle, if it will be asked, these are leaf-shaped glands, and these are actually present at the back side of the urinary bladder. That means towards the vertebral column it is present. It is not present at the front of the urinary bladder, and these are leaf-shaped. And keep it in your mind, these glands are opening into the ejaculatory duct. Okay, so here in this specific figure you can see these are the leaf-shaped glands. This gland and this gland I am talking about, this one and this one, these are commonly leaf-shaped gland and present at the back side of the bladder, and these are the seminal vesicles, and which are secreting their chemical within the ejaculatory duct. Okay, next, prostate gland. What is about this gland? Prostate gland is secreting remaining 20 to 30% of the total volume of the semen. Keep it in your mind, the secretion which is secreted by the prostate gland, that is acidic in nature, and it is present in the junctional region of the urinary bladder and ejaculatory duct. From the figure also you can see properly if this is the urinary bladder and if this is the ejaculatory duct. So in the junction of them there is a heart-shaped, rather if you consider both of the glands from both sides, they are actually heart-shaped gland-like structure, glandular structure is forming, and that is known as prostate gland. Okay, next, after prostate gland there is another gland is also present, which is also another accessory gland, and that is known as Cowper's gland or bulbourethral gland. Keep it in your mind, this is a mucus secreting gland, and that is acting as a lubricant for the sperm movement. Alkaline fluid secreted by this specific gland, and it is neutralizing the acidic medium within the male reproductive tract, specifically within the urethra, as well as it is also neutralizing the acidic medium within the female reproductive tract when the sperms are moving or going towards their destination, that means up to ovum. So here you can see the small two glands, very small, these two glands which are present just below the prostate gland. These two are known as the Cowper's gland or bulbourethral gland, and they are secreting the alkaline fluid, and they are actually acting as a, their secretion is acting as a lubricant, and they are also secreting the mucus all. Okay, so this is overall the three kinds of male reproductive glands: seminal vesicle, which is constituting the maximum portion of the semen, then prostate gland, which is constituting the minute portion of the semen, and at last the Cowper's or bulbourethral gland. Okay, so we have understood one thing, that the function of these three glands are only to produce the semen in a proper quantity, so that the sperm will be getting nourishment as well as the sperm can move up to its destination in a proper way. Next structure is coming, and the last structure under the male reproductive system, that is the male external genitalia or the penis. What is that? Male external genitalia structure you can see here. This is the longitudinal section of the male external genitalia, and this is the transverse section of the male external genitalia. So let's see the structure so that it will be very much clear to us. First of all, within the male external genitalia, there is a presence of a canal-like structure or the tube-like structure that is known as urethra. And this tubular part passing through the length of the male external genitalia, because after the ejaculatory duct, the next tubular structure, if it is present anything, that is urethra. And the urethra has two portions. One is the canal-like portion, which is known as urethral meatus, and then there is another aperture through which the urethra is opening outside, that is known as urethral orifice. Already we know about this, and again I am showing you in this figure. So this is the male external genitalia portion, and the canal which is passing through it, this is the canal portion, that is known as urethral meatus, and when it is opening outside through a aperture, that is known as urethral orifice. Okay. Next, there are two muscular tissue or connective tissue are present, which are actually providing the strength of the male external genitalia, or rather it is responsible for the erection of the male external genitalia, that is corpora spongiosum and corpora cavernosum. Which are there? This is the muscular connective tissue, the corpora spongiosum, and surrounding the urethra. That means if this is the urethra, that is surrounded by the muscular tissue, and that is known as corpus spongiosum. Corpus spongiosum or corpus spongiosum, that is actually this specific tissue. You can see here the little bit light red color. This is surrounding the urethral meatus, and that is known as corpus spongiosum or corpus spongiosum. Next, this corpus spongiosum is again surrounded by another muscular connective tissue. You can see here the little bit blue colored. So this specific portion, whatever I am showing here, which is covering the corpora spongiosum or corpus spongiosum, that is known as corpora cavernosum. Okay. This is another ligamentous muscular tissue which is surrounding the corpora spongiosum, and exclusively keep it in your mind, these two muscular ligamentous connective tissue layer are exclusively present for erection of male external genitalia or penis. Next, artery is also there. Artery is actually present for the distribution of the blood to the inner portion of this specific tissues, corpora cavernosum and corpora spongiosum. In this specific figure, you can see properly the arteries. This is the deep artery, this is the deep artery, and this is the deep artery. Okay, and these are the dorsal blood vessels which are coming from the dorsal surface of the male external genitalia. This is the transverse sectional view, and this portion, purple color, these portions are corpora cavernosum. Okay, and this is the urethra which is surrounded by the corpora spongiosum. So keep it in your mind, this is the urethral aperture which is surrounded by the pink color corpora spongiosum. It is very much clear in this diagram, and this is the entire portion which is actually made up of corpora cavernosum, and in the centrally located portion of the corpora cavernosum there is a presence of the deep artery. Okay, and through the artery, when the blood will flow to the inner part of the male external genitalia, obviously it will help with the erection. Glans penis, what is that? That is a fleshy outgrowth at the apex of the external genitalia. So if you consider the male external genitalia, this specific fleshy portion I am talking about, this fleshy portion which is little bit bulged, and that is known as glans penis. Rather, it is surrounding the last portion of the urethral meatus, and when it is opening into the urethral orifice. Next, skin is also there, that is covering the external genitalia from outside, that is a skin layer. And prepuce, what is prepuce? If you see the anterior or apex portion of the male external genitalia, it is containing the folded skin, so that the glans penis will be get protected in a proper manner. So the folded skin which is present in the apical portion of the male external genitalia, that is commonly known as prepuce. The prepuce structure is not shown here actually. The skin has actually the skin is also not shown here properly, but keep it in your mind, this male external genitalia, whatever the structure you can see, that is covered externally by the skin as a protection, and in the anterior portion that skin is folded so much so that the glans penis also get protection from any kind of external shock or infection, any kind. So this is overall the structure of the male external genitalia. I think the male external genitalia structure, male external genitalia structure, and whatever the structural parts are present, I think it is very much clear to each and every one. Now we are moving for the next thing, and that is obviously it is not the structural part of the male reproductive system. this is the mechanism oriented portion, and here we will be discussing about the mechanism of sperm formation within the seminiferous tubule. And keep it in your mind, this process is known as spermatogenesis, that is the production of the sperm within the seminiferous tubule. So let's see the procedure of the spermatogenesis. First of all, you can see here the spermatogenesis is starting from the germinal epithelial cells. Which are germinal epithelial cells? The peripheral cells which are present in the seminiferous tubule. That means if anybody asks you whatever the outer layer or outer covering membrane of the seminiferous tubule, obviously our answer will be coming germinal epithelial cells, because these germinal epithelial cells are only the outermost layer or the covering layer of the each and every seminiferous tubule. So these germinal epithelial cells, they will be dividing mitotically, because the mitosis cell division takes place, and by mitosis cell division they will be producing type A spermatogonial cells. Keep it in your mind, after mitosis cell division that will be producing type A spermatogonial cells. These type A spermatogonial cells will be again proceeding through the another mitotic division, and it will be forming the type B spermatogonial cells. So first of all, the spermatogenesis process is starting from the outer membrane of the seminiferous tubule, that is germinal epithelial cells. Those cells are dividing mitotically to produce the type A spermatogonial cells, and next those type A spermatogonial cells are further again dividing to form the type B spermatogonial cells. Type B spermatogonial cells, up to formation of this is entirely coming within the multiplication phase, because we can understand from single germinal epithelial cells so many type B spermatogonial cells are taking birth. Actually 4 spermatogonial cells are taking birth from a single germinal epithelial cells. That means the number of cells are getting 4 times. So that's why this is commonly known as multiplication phase. Next, type B spermatogonial cell is entering within the growth phase, where due to presence of some growth factors within the spermatogonial cell, only their size is getting bulged, and that is known as growth phase. In this phase, what is happening? The type B spermatogonial cells will be getting converted into primary spermatocyte cells. And keep it in your mind, the growth is happening in this phase, that's why the phase name has been given growth phase. Now, primary spermatocyte which has been produced, that will be entering within the next cell division, but that cell division is not mitosis, rather it is the meiosis, because the chromosome number of the primary spermatocyte, it will be getting half now. So the primary spermatocyte, it is obviously up to primary spermatocyte, keep it in your mind, the number of chromosome is present 12, but just the primary spermatocyte will enter within the meiosis 1 cell division. Automatically it will be producing the secondary spermatocyte cells, and these cells are exclusively containing haploid number of chromosome or n number of chromosome. This haploid number of chromosome containing secondary spermatocyte will be again entering to the next phase of the meiosis, that is the meiosis 2. And just it will be passing through the meiosis 2, it will be forming the next phase, that is the spermatid. Keep it in your mind, spermatid is also a haploid number of chromosome containing cell, or rather it can be told as the precursor of the spermatozoa, because after spermatid formation, when it has been done, after that there is one step is present, or the one mechanism is present, that is known as spermiogenesis. Up to spermatid formation you can consider it as a spermatogenesis, but after spermatid formation the next mechanism which is taking place, that is the spermiogenesis. Why? Because in the spermatic structure, spermatid is a round shaped structure, within that the nucleus is also round, no such kind of sperm like a spermatozoa like structure found in case of spermatid, but the modification of the spermatid into spermatozoa, how it is happening, that is actually happening by the help of the process, that is spermiogenesis. So the last step, that is the spermiogenesis and forming the mature spermatozoa. Okay, so this entire 3 steps starting from the primary spermatocyte, that is under the maturation phase, because the maturation or the formation of the spermatozoa is taking place. What is spermiogenesis and what is happening in the spermiogenesis, that we will be discussing in the next slide. But before going to the next slide, here I want to say that there are 3 phases within the spermatogenesis: multiplication phase, growth phase and maturation phase, and these are the steps. Please keep it in your mind, these steps in a sequential manner, and try to represent this mechanism also in the exam like this flowchart manner, so that it will be very much lucrative. Okay, so now we are going for the next slide. In the next slide, it is actually shown properly the spermatogenesis process by showing the number of chromosomes. This is the primary germ cell or the germinal epithelial cell, which is dividing by mitosis cell division and forming 2 type A spermatogonial cells. Then type A spermatogonial cells are dividing again to 4 spermatogonial cells. Those are type B spermatogonial cells. Those are all diploid number of chromosome containing. Suppose from any of the type B spermatogonial cell, the primary spermatocyte is taking birth just by increasing their size. That's why it is under the growth phase. Now the primary spermatocyte will be entering within the first maturation division, that is the meiosis 1, to form haploid number of chromosome containing secondary spermatocyte. These secondary spermatocytes are entering again within the second maturation division or the secondary maturation division or the meiosis 2 and forming the 4 spermatids, and these spermatids will be entering within the spermiogenesis process and they will be developing, they will be just changing, converting their structure into mature sperm. So this is overall the spermatogenesis process, whatever I have told in the previous slide. In the previous slide only I have written the name. Here the numbers of the chromosomes are also mentioned. And how they are happening, this is actually the figure. This is our germinal epithelial cell layer. From there the spermatogonial cells, okay, and from the spermatogonial cell primary spermatocytes have came, then secondary spermatocytes have been produced. These are the spermatids, and these spermatids have produced these flagella containing tadpole shaped spermatozoa or sperm. So this is actually the figure how it is happening within the seminiferous tubule. The small portion of the seminiferous tubule has been shown here. So this is overall completed. Next we are going for the spermiogenesis. What is about the spermiogenesis? This is actually the process of the spermiogenesis. Here you can see this is the spermatid structure, how this spermatid is getting developed into the tadpole shaped spermatozoa, that is actually shown here. So keep it in your mind, this spermatid is initially rounded shaped, the nucleus is also rounded. So the change of various cell organelles, change in the shape of the spermatid, all are coming, and afterwards approximately after 24 days this sperm structure is producing. Okay, so let's see whatever the changes are coming. The changes which are coming after spermiogenesis, during the spermiogenesis, that is the nucleus that is becomes scimitar shaped. Scimitar shape means I am talking about, suppose within the spermatid the sperm nucleus is present in a round shaped manner, but when it is actually elongating, the nucleus is elongating and forming a conical structure, then that structure is known as scimitar shape. You can see here the nucleus is rounded, but you can see here the elongation of the nucleus is happening, and after that the nucleus is getting too much elongated and conical shape, and that shape is commonly known as the scimitar shape. Okay, Golgi bodies which are present within the spermatid, that will be forming the acrosomal cap. Keep it in your mind, this is the Golgi body which is present in the adherence portion of the nucleus, and this specific black color Golgi body portion, it will start covering the anterior portion of the nucleus, and this specific Golgi body will be forming further a cap-like structure or a protective covering that is known as acrosomal cap, and it is containing a sac-like structure that is acrosomal vesicle, because this acrosomal vesicle is responsible for producing the enzymes which are responsible for rupturing the layers surrounding the ovum. Okay, mitochondria which is present within the spermatid, mitochondria is actually further producing the nebenkern. Nebenkern is actually a sheath which is protecting the axoneme, rather the central axis of the sperm, and this specific nebenkern is exclusively containing so many mitochondria, and they are protective covering of the central axis, as well as it is also providing the energy during the sperm movement. Next, cytoplasm. Cytoplasm is actually the cytoplasmic portion which is present within the spermatid, that will be producing another layer that is commonly known as manchitti, and this manchitti layer will be surrounding the nuclear portion, and entirely in the head portion the nuclear portion is entirely surrounded by the manchitti, as well as in the middle piece portion also the nebenkern is surrounded by the help of manchitti. These specific structures where they are actually present, that I will be showing in the next slide in the structure of the sperm. Centriole will be present in the spermatid, and those centrioles are actually coming towards the downward. This is the centriole, and this centriole, why it is come downward, so that it will be producing this flagellum, the tail like structure, and which will be giving the tadpole shape to the sperm. So this is overall the changes present or happening within the during the spermiogenesis process. Okay, and after getting this spermiogenesis process over, automatically what will happen? The sperm will be coming out from that seminiferous tubule, and that process is already called as, or I have told the name, that is spermiation. Now we are going for the structure of the sperm. This is the structure of the sperm. Keep it in your mind, the sperm is subdivided into three portions. One is head, second one middle piece, and another one is tail. Sometimes neck portion also can be included, but mainly three portions: head, middle piece and tail. Now let's see whatever the portions are present within the head. At the apical portion of the head, there is a presence of acrosomal vesicle. This is the acrosomal vesicle which is produced from the Golgi body, and this acrosomal vesicle is containing so many hydrolytic enzymes, so that those enzymes can perforate the outer coverings of the ovum, okay, during the fertilization process. After that, there is a conical shaped nucleus, that is the scimitar shaped nucleus present. Just below the nucleus, there are two types of centrioles are present, and one is proximal centriole, another one is distal centriole. Out of these two centrioles, the distal centriole is responsible for producing the axis. This is the axis I am showing you in the figure. This is the axis that is commonly known as axonym. This axonym or the central axis is produced by the distal centriole. Keep it in your mind. And keep it in your mind, in the middle piece region, this central axis or the axonym is surrounded by a membrane, and this membrane is present in a zigzag manner, and this membrane, whatever I am showing in the figure, that is commonly known as mitochondrial membrane, or it is biologically it is known as Nebenkorn. Another most important point is that the cytoplasmic portion which was present within the spermatid, that cytoplasmic portion is now present surrounding this nucleus portion as well as surrounding this Nebenkorn portion. So this is the pink color portion, that is the cytoplasm, and keep it in your mind, the cytoplasmic portion name we have given, that is Manchete. And after that, there is a filamentous sheath is present just covering the central axis, which is also present within the tail or flagellum. So this is the flagellum portion, that is the last part of the sperm, and that is helping in the sperm motility. Just see the structure. The head contains the nucleus and the enzyme filled acrosome. Acrosomal enzymes allow the sperm to penetrate the egg's barriers, or that is the egg's covering. The neck encloses a centriole. The centriole will fuse with the second centriole contributed by the egg. Obviously there are normally within one cell one centriole will be present, but your question can come, why the two centrioles have come here? The reason is one centriole is itself of the sperm's centriole, and another one contributed by the egg to form the centrosome. Mid piece or middle piece is packed with the mitochondria, that is commonly known as Nebenkorn, which produces ATP, which is necessary for the movement of the sperm. And the tail has a flagellum that acts as a propeller. Okay, and another one more thing here, it is not mentioned, that is the cytoplasmic part, which is covering the head portion, middle piece portion and tail portion entirely, and that is commonly known as Manchete. Clear? Now we have actually reached to the last portion of the male reproductive system, that is hormonal regulation in the male reproductive system. So let's see about the hormonal regulation. What about the hormonal regulation? Keep it in your mind, always it is getting controlled by the hypothalamus, because it is considered as a master of master gland. That's why hypothalamus is releasing the releasing hormone, that is the gonadotrophic releasing hormone, GnRH. It is coming to the anterior pituitary. GnRH can be of two types, FSH-RH and LH-RH. That means follicle stimulating hormone releasing hormone and luteinizing hormones releasing hormone. LH-RH and FSH-RH, these are the two hormones under GnRH. So they are coming to the anterior pituitary, stimulating those specific cells, okay, present within the anterior pituitary, and after getting stimulated those cells, obviously from there FSH is coming out and LH is coming out, and FSH and LH both are coming up to the testis. FSH is stimulating the spermatogenesis process, as well as FSH is stimulating the Sertoli cells to facilitate their secretion, because keep it in your mind, this Sertoli cell, whatever I have discussed in the structure of the seminiferous tubule, that is a conical shaped cell, and that is providing the nourishment to the spermatozoa. Okay, and sometimes this Sertoli cell is also producing a hormone, that is inhibin, that is also responsible for inhibiting the process of the spermatogenesis. That is also important, that it's not mentioned in these slides, but you can keep it in your mind that the Sertoli cell is providing the nourishment to the growing spermatozoa within the seminiferous tubule, as well as they are also releasing the inhibin, which can inhibit the spermatogenesis process. But Sertoli cell again facilitates the spermatogenesis process also, and that is controlled by the hormone FSH, that is the follicular stimulating hormone. Next, LH also, which has come from the anterior pituitary, which is also called as ICSH, that is interstitial cell stimulating hormone, because it is released from the Leydig cell, that is still stimulating really the Leydig cells, so that from the Leydig cell the testosterone hormone can release. Okay, now here it is actually shown that anterior pituitary, from there when the FSH and LH both are coming out, that means the anterior pituitary has got stimulated by the GnRH. So obviously these two green color arrows, we can consider them as a positive feedback control or positive feedback mechanism. But when the huge quantity of testosterone has been produced and they have come to the blood, as well as when the Sertoli cell has produced so much amount of inhibin, because inhibin is written here, whatever just I have told, so automatically this inhibin will be working on the anterior pituitary, as well as testosterone will be working on anterior pituitary and hypothalamus, so that further the gonadotrophic releasing hormone as well as FSH and LH from the anterior pituitary, it will not release, because FSH and LH, whatever the functions are performed by them, already those have been done. So when those processes or those mechanisms have been already completed, now there is no necessary to release of the FSH and LH further from the anterior pituitary, as well as there is no need of release of the FSHRH and LHRH from the hypothalamus. So for that reason, the negative feedback control mechanism is stopping or inhibiting the secretion of FSH and LH from the anterior pituitary, and also inhibiting the secretion of GnRH from the hypothalamus. So these red arrows will be actually considering the negative feedback control. Clear? So another more important thing is that here also one green arrow is given, what is actually showing that Leydig cells which are secreting the testosterone, which is the male sex hormone, that is also controlling the Sertoli cells for spermatogenesis process. Okay, so this is entirely the hormonal control or feedback mechanism control within the male reproductive system. So up to this slide we have completed, and up to this slide when we have completed, the entire male reproductive system is completed. So I think you have understood each and every portion of the male reproductive system, and I think you have enjoyed this entire video a lot. Maybe this video is little bit lengthy, but entire male reproductive system has to be finished in one video. That's why I have completed this video by taking extra time also. So you will be just studying one by one slide in a proper manner, and information has to be kept properly, because these informations are very much necessary, and each and every informations are very much important. So thank you everyone for listening. Okay, thank you.